Hi, everyone. Me again, the disembodied head of your professor, talking to you through the internet. So this lecture, we're going to talk about the gene regulation portion of this class. We focus on chapter 14 for this, and specifically, we only focus on the first two parts, because trust me, that's enough. So this first lecture is going to discuss a general overview of gene regulation, why it's important. And then we'll get a little bit into the regulation of the Lac Operon, and we'll finish that off in the second part of this mini lecture. So to begin with, just a reminder to let you know what gene regulation actually is. So gene expression is the process that we're regulating in this case. Gene expression, remember, is the transfer. It's basically the central dogma of molecular biology, right? The transfer of information from DNA through transcription to mRNA, through translation to proteins. That process is called gene expression. And we're talking about the regulation of that process in today's lecture. So why is it important to regulate this process in cells? So what this means is that this process of gene regulation, this process of gene expression, can be varied under different cellular conditions. And that's really important. Now, it's also important to let you guys know that some genes are not regulated at all. So some genes are called constitutive. These genes have constant levels of expression, so that protein from these genes are made continuously all the time at roughly the same level. As you might expect, this includes a lot of what we would call housekeeping genes. In other words, genes where you require the gene product or the protein all the time. So one example might be a gene that's required for cellular respiration. So a gene that's important for your mitochondria to work properly, right? You need mitochondria in every cell working all of the time. So the kind of proteins that are involved in cellular respiration are constitutively expressed. Why regulate gene expression at all then? Well, it's because you don't need every gene making every protein all of the time. In fact, if you did that, it would be a great big waste of energy, right? So gene expression allows you to produce proteins in the right amounts when they are required. And it basically leads your cells to not wasting energy and making proteins that you don't need. Now, we're going to talk specifically about gene regulation in bacteria today. Now, gene regulation in bacteria is simpler than eukaryotes, but a lot of the same concepts are used in the way that bacteria regulate their genes compared to the way eukaryotes do. And essentially, the way I always think about gene regulation is, if you can think of a place to regulate gene expression, it probably is regulated at that place. So some examples are shown on the right-hand side of this slide. Transcription regulation is really important. The decision to turn a gene on or not, that's pretty fundamental to gene expression. And then even after the mRNA is made, you can regulate the translation of that mRNA. Some can be regulated, some can be translated into proteins in different amounts. My dog just joined me. I was like, who just came in? And even after the protein is made, things like the stability of the protein can be regulated, and things like that. So you can regulate basically the amount and the activity of the final protein product in lots of different ways in cells. So there's lots of different ways, as shown here, that you can regulate gene expression. Mostly today, we're going to talk about transcriptional regulation, because that's very fundamental, right? The idea whether or not the cell makes the decision to transcribe a gene is a big one for a cell. So in prokaryotes, A lot of gene expression is regulated at the level of transcription, specifically transcription initiation. We can talk about that today. In eukaryotes, there's a much more complex there are much more complex ways that they regulate their genes, which makes sense, right? We're a multicellular organism. Each of our cells has specific jobs. Gene regulation, very important and must be closely monitored and controlled. So what we're going to talk about is some sort of fundamentals of transcription regulation today, or gene expression regulation. And basically, conceptually, you can have two general regulatory mechanisms. You can have an inducible mechanism or a repressible mechanism. What does that mean? Well, the clue is in the name here. So if you are a gene that is inducible, it means that you're normally off, but you're capable of being activated or turned on. You are inducible, right? The other type of system that one could imagine is a repressible system, where the opposite is true. In a repressible system, it's on, usually, but capable of being repressed, capable of being turned off. Now, it's generally true that if you're an inducible system, you're used for, at least in bacteria, you're used for something that's important for catabolism, breaking down things. And if you're usually on, you're normally on because the cell really needs whatever it is you do. So you're normally building something or making something. And here's the, oh, thank you. Thanks, honey. Can I have a word? Yeah. Thank you. Cup of tea in my DNA mug, very important. Requested a Werther's today. Need a bit of old man candy. So when you are talking about inducible systems, here's how they kind of work. As I said, they're usually off. Thank you. You can do that in a couple of different ways. Here's an example of a piece of DNA. Note that there's a promoter here. We've talked about that this semester a little bit. The promoter is where RNA polymerase binds to transcribe a gene. So inducible systems, which are usually off, sometimes that's because there's a repressor protein that is bound to the DNA, as is shown here. Notice that that's blocking the promoter and preventing RNA polymerase binding and transcribing a gene. So this is a negative regulatory state. How does it go from being off to being on? So off state is on the left, on state is on the right. What you can see has happened in this case is that the repressor protein has been bound by some kind of regulatory molecule called an inducer. It's an inducible system. That caused this repressor to fall off the DNA, allowing RNA polymerase to bind and transcribe this gene to make mRNA. So that's one way you could do an inducible system. The other way you can do it is what is called positive regulation, which means that there's essentially no repressor involved. So this gene is just off naturally all of the time. In order to be turned on, it has to be bound by some kind of activator protein, as is shown in the bottom panel here. And what you can see here is that the opposite is true, right? This is positive regulation. So an activator does not bind unless it's somehow induced to bind by a small inducer molecule, which now causes it to bind to DNA and allow transcription of this gene. So both of these situations, negative regulation and positive regulation, can be used in inducible systems because they're just ways that you keep a gene off and then decide to turn it on, right? Note that in both cases, the binding of some important inducer molecule is required to turn the gene on. And what that inducer is doing in both of these cases is causing some kind of three-dimensional conformational change in the protein, which causes it to either fall off the DNA, as is the case with this repressor protein, or in the case of the activator protein, the change in conformation causes it to bind to a specific DNA sequence and recruit RNA polymerase. So that's inducible systems. What about these repressible systems that are usually on, but sometimes turned off? So in this case, the on state is going to be on the left and the off state is going to be on the right. What you see here is sort of a mirror image of what you saw in the previous slide. So they're normally on, right? So here's the on state on the left, here's RNA polymerase doing its thing, making its mRNA. Here we have a repressor protein that is floating around, not repressing the gene at the moment. And you'll never guess what's going to happen. It's going to bind some kind of co-repressor. That co-repressor, when it binds to this protein, causes a conformational change, which causes it to bind to DNA and block the promoter from binding RNA polymerase. That's negative regulation. A similar idea can happen in repressible systems as does in inducible systems, where it comes to positive regulation as well. That is, an activator is bound, normally bound to this protein, recruiting RNA polymerase, allowing it to transcribe this gene. And then in this case, a co-repressor or an inhibitor is actually it's an inhibitor, not a co-repressor, because it's binding an activator protein. So an inhibitor will bind to this activator protein, causing it to fall off the DNA, causing it to no longer recruit RNA polymerase, turning off transcription of the gene. So these are all transcriptional regulatory mechanisms that are used very commonly in lots of different genes. Just depends on the gene, whether it's either, active, whether it's either normally off, in other words, an inducible system, or normally on, in other words, a repressible system. And both of these systems, both inducible and repressible, can be either negatively or positively regulated. I know that sounds confusing, but here's how I ground myself when I'm thinking about these things. So if I go back a couple slides, I basically, when I'm addressing systems like this, I just think about what the names mean. So if a system is inducible, it means it's usually off. What does that mean? It means it's either bound to a repressor all the time, or it is not bound to an activator. Conversely, with repressible systems, they're repressible, right? So that means they're on most of the time. So they're either not bound to a repressor that can bind to shut them off, or they are constantly bound to an activator, which can be removed. So the way I ground myself in these terms is by thinking about what the terms actually mean, and that's helpful, I think. So what we're going to do in the rest of this lecture and the entirety of the next lecture is examine the Lac Operon and what it does. So at the start of the 20th century, the observation was made that in bacteria, if a cell needs a particular enzyme, then that enzyme all of a sudden appears when its substrate is presented to the cell, right? This idea was termed enzyme adaptation, because it's like the cell is adapting by making these enzymes. And that's exactly what we're going to look at today when we look at the Lac Operon. And in the case of the Lac Operon, what it actually does, so the Lac Operon is a piece of DNA that's going to make the enzymes, or contains within it the instructions to make the enzymes. Remember, enzymes are just proteins. to digest lactose. That's why it's called a Lac Operon. So think of it like this. You have millions of E. coli bacteria living in your gut right now. Most of the time, no one likes to think about the contents of their own gut too much, right? But most of the time, those cells in your intestines are not being exposed to lactose unless your diet consists entirely of dairy, which is just not a good idea. So when I drink this cup of tea, for example, there's some milk in it. When that milk gets down to the bacteria in my gut, all of a sudden, there's some lactose in that milk, which is a sugar, and they can use that for metabolizing to give them energy. But they don't want to express the digestive enzymes, their own metabolic enzymes for lactose all of the time, because, I mean, I'm English, I drink quite a lot of tea, but I don't drink it 24/7, so there's not always lactose present in my digestive tract. And so that's what we're talking about when we talk about the Lac Operon. We're talking about the fact that when a cell is exposed to lactose, all of a sudden it makes the enzymes that it needs to digest that lactose. Because of this, it makes sense that it is an inducible system, right? It's normally off, but it can be induced when lactose is around. So one more term I want to introduce to you and talk about is that what does the word operon mean? It's pretty simple. So an operon so operons are mostly present in bacteria. There are a few in the human genome too. They're quite prevalent in the bacterial world though. And basically all they are two or more genes that are under the transcriptional control of a single promoter. And normally those genes that are under the control of a single promoter are involved in a single process, which makes sense. So in the case of the Lac Operon, what we're talking about are three genes that are all under the transcriptional control of one promoter. So they're all turned on at exactly the same time, and they're all used to metabolize lactose, right? So what that means is if I'm a cell and someone needs to be, and I live in your gut and you eat a big piece of cheese, right? All of a sudden there's loads of lactose in my gut. I need these three enzymes that are going to digest it for me, and I need them all at the same time because that's what they're used for. So they're organized in this very nice way where you turn them all on at the same time because you only ever need them all at the same time. And this allows for a very rapid transcriptional response and allows cells to respond to their environment in dynamic ways. So here is the piece of DNA that is the Lac Operon. And right now we're just going to focus on the three genes that are involved in the Lac Operon. Their names are LAC-Z, LAC-Y, and LAC-A. The first of these, and the biggest, is on the left here. This is called LAC-Z. And so remember, this is just a piece of DNA that contains these three genes. The first one encodes an enzyme called beta-galactosidase. This is involved in cleaving lactose, the sugar. And what that does, as you'll see in a second, lactose is basically two sugar rings attached together. This just cuts them in half so they can be used by the cell for energy. The second of the three genes is LAC-Y. That is called lactose permease. And again, the clue is in the name here. Lactose permease is a protein that basically opens up the cell membrane to allow lactose into the cell. Sugars don't normally pass through cell membranes very easily because they're hydrophilic. And so they need some kind of special channel to allow them to get in. That's what permease is. It allows lactose into the cell. The third gene shown in this kind of burgundy color is something called galactosidase transacetylase. You don't need to worry about that because amazingly, this Operon has been studied for like 100 years. And no one really knows what the transacetylase is for. So you don't really need to worry about that. But it is part of the operon, and so we have to talk about it. So what that means is, for a bacterial cell to use lactose as an energy source, it needs two of these three proteins. It needs Lac-Z to chop up the sugar into usable pieces, and it needs Lac-Y to let the sugar into the cell in the first place. And so these are the functional parts of this operon as regards making proteins. When this operon is activated, RNA polymerase will bind at the promoter, which is shown in green here, and it will proceed down this piece of DNA, transcribing it into this long mRNA. And this is what I'm getting at when I talk about operons. There's three genes here, and they're transcribed under the control of the same promoter. And in fact, they're all on the same piece of messenger RNA. So with this piece of messenger RNA, there's one start codon here. This is your ATG. So when a ribosome binds this to translate it, it will start translating at this start codon, and then it will make this protein, which is beta-galactosidase. And then when it gets to the stop codon, that protein will be released from the ribosome, but the ribosome will stick around on the mRNA, and it will immediately encounter another start codon for the second gene, Lac-Y. And the same thing will happen. It will go ahead and make permease, and then after that, it will make this third protein, transacetylase. This piece of RNA has a special name. It's called polycistronic. A cistron is like the really old name for a gene, and polycistronic just means multiple genes in one mRNA. And again, this is because all of these proteins are important for the cell to use lactose, especially these first two. So they're all made at the same time, and the cell can react quickly when you drink that big glass of milk, and the bacteria in your gut can then digest it for their own use. All right, so here is what that looks like if I am a bacterial cell. This big oval is meant to show a single bacterial cell. And what you see here is lactose permease. So like I said, this is the gateway for lactose to get into the cell. Once the lactose gets into the cell through this little channel, this is what it looks like. It's this two-ring sugar. Beta-galactosidase is then going to cleave it into galactose and glucose, which are the component parts of lactose. And they will be used in the cell for energy. Beta-galactosidase has this little side reaction that it also catalyzes that makes this weird product called allolactose that we'll talk about in a minute. But beta-galactosidase, its main role is to cleave lactose into two parts, glucose and galactose, for use in the cell as energy. Okay, so there are three genes involved in the Lac operon itself: Lac-Z, Lac-Y, and Lac-A. There is a fourth gene that is not technically part of the operon, but is essential for its function. And that is a gene that's shown on the left here. It's called a regulatory gene. It's called Lac-I. And what it encodes is a repressor protein. So here it is on the left. This regulatory gene makes a repressor protein. But notice that it's not part of the Lac operon itself, which is separate from the regulatory gene. Very importantly, the Lac-I gene product, the Lac repressor, is constitutively expressed, which means that this gene is always on, just making Lac repressor constantly. And that's going to become important when we talk about the regulation of this operon. Okay, So we've talked about 4 genes now, the 3 involved in the Lac Operon and the Lac repressor, which is not technically part of the Lac Operon, but very important for its regulation. Within the Lac Operon itself, there are 3 pieces of DNA that don't make a protein, but they're really important for the regulation of the Lac Operon. And those 3 pieces of DNA are called, well, I'll go through them in order. One is the Lac promoter, or Lac-P for short. That's the sort of cream one in the middle. This is a promoter. You guys know what that does. It binds RNA polymerase to allow it to transcribe the operon. All 3 of these sites are essentially just special sequences in the DNA that are landing sites for DNA binding proteins. One of those is RNA polymerase. Lac-O on the right here in green is called the operator sequence. This actually binds to the Lac-I repressor. So the product made by the Lac-I gene, the protein it makes, will bind to this sequence, the operator, to repress the transcription of the operon. Last but not least is something called the cap site, which stands for the cAMP binding site. And we will talk about that as we get through to the sort of end part of the next lecture. But again, it's just a landing site, a special DNA sequence that binds to a DNA binding protein. All right, so finally, this is the sort of end of the introduction to the Lac Operon. So the Lac Operon itself is shown by this line right here. It goes from, it includes these 3 regulatory sequences and these 3 functional genes. It also includes something we're not really going to talk about, the Lac terminator, which just tells RNA polymerase where to stop. So really, if you want to separate the Lac Operon into its component parts, you would say that it has 3 structural genes that are going to encode enzymes that are important for lactose metabolism. And it contains 3 regulatory regions, which are just DNA sequences that bind to different proteins and are involved in transcriptional regulation of the Lac Operon. All right. So that's at the end of the intro. Please watch the next video because it will include not only how this thing works, but also some tips about how to address some of the questions on this, this week's assignment. So I'm going to go ahead and start that video right after I eat this delicious Werther's original. So I will see you then. Thank you.